The nervous system has two different parts which play an important role in its functioning. The two parts are the central nervous system and the peripheral nervous system. The CNS consists of a brain and a spinal cord, which serve as the nervous system's command station. The PNS is everything else. The PNS contains nerves, which leave the brain and the spinal cord and travel to the necessary body parts. The main job of the PNS is to send information received by the sensory receptors to the CNS as quick as possible. Once the CNS has made a decision, the PNS relays the orders back to the body. Information is carried to and from the brain by nerves. These nerves are called cranial nerves. Information is carried from the spine by the spinal nerves. The PNS has two important parts, the motor division and the sensory division. The sensory division collects impulses from the sensory receptors in the organs and carries impulses to the CNS. However, the motor division collects messages from the CNS and delivers to the right body organ. The main two differences between the CNS and the PNS is that the CNS processes information and enacts involuntary communication, whereas the PNS integrates received information and carries out voluntary communication.
A mammalian neuron is made up of 6 different parts. These are the cell body, dendrons, an axon, Schwann cells, a myelin sheath, and the nodes of Ranvier. A cell body contains a nucleus and large amounts of rough endoplasmic reticulum. This is associated with the production of proteins and neurotransmitters. Dendrons are small extensions of the cell body which subdivide into smaller branched fibers called dendrites. These carry the nerve impulses towards the cell body. An axon is a single long fiber that carries nerve impulses away from the cell body. Schwann cells surround the axon, protecting it and providing electrical insulation. They also carry out phagocytosis, which is the removal of cell debris, and play a part in the nerve regeneration. Schwann cells wrap themselves around the axon many times so that layers of their membranes build up around it. A myelin sheath which forms a covering to the axon and is made up of the membranes of the Schwann cells. These membranes are rich in a lipid known as myelin. Neurons with a myelin sheath are called myelinated neurons. Some neurons lack a myelin sheath and are called unmyelinated neurons. Myelinated neurons transmit nerve impulses faster than unmyelinated neurons. Nodes of Ranvier are gaps between adjacent Schwann cells where there is no myelin sheath. The gaps are 2 to 3 micrometers long and occur every 1 to 3 millimeters in humans.
When a neuron is at rest, it is not transmitting an action potential. In fact, it is actively transporting ions across its cell surface membrane. Sodium-potassium ion pumps use ATP to pump 3 sodium ions out of the cell and 2 potassium ions into the cell. The plasma membrane is more permeable to potassium ions than to sodium ions, so many diffuse out again. The cell cytoplasm also contains large organic anions which are negatively charged. The interior of the cell has a negative potential compared to the outside. The cell membrane is polarized and the resting potential is about minus 60 millivolts.
In order for a signal to be passed along, the neuron must initiate an action potential. This is made up of a number of steps. As an action potential is achieved, the voltage or potential difference across the membrane changes, due to the change in the number of sodium ions inside the neuron. Before the transmission occurs, the membrane is at minus 60 millivolts. This is the resting state and the inside of the cell is described as polarized. Sodium channels open and ions begin to diffuse into the cell, causing it to become depolarized. This means that it is less negative with respect to the outside of the cell. In order for the transmission to continue, the potential across the membrane must meet minus 50 millivolts. This is known as the threshold potential, and it determines whether or not an action potential will be created. This is why reaching action potential is also known as an all-or-nothing process. However, if the threshold potential is achieved, then the voltage-gated sodium ion channels will open, allowing more ions to flood in. The potential difference reaches approximately positive 40 millivolts, before the sodium ion channels close and potassium ion channels open. Potassium ions diffuse out of the cell, repolarizing the potential difference. However, this overshoots slightly in a process called hyperpolarization. Afterward, the resting potential is restored by sodium-potassium ion pumps.
Neurons are designed for action potential conduction. They help meet the functions of the nervous system. For example, Axon terminals store neuron transmitters which transport impulses through the affected tissue. The axon is covered by layers of myelin sheath which act as a shield for the axon, as well as increasing the speed of nerve impulse conduction. The unmyelinated spaces seen on the axon are called nodes of Ranvier. This is where sodium and potassium channels are situated. Here, the action potential is able to jump from one node to another through the exoplasm in a saltatory conduction. Here, the speed of conduction increases. However, in the unmyelinated fibers, myelin is not produced. This allows the action potential to move down the unmyelinated axon in a continuous conduction, lowering the speed of conduction.
The arrival of an action potential at the end of the presynaptic neuron causes calcium ion channels to open and calcium ions enter the synaptic knob. The influx of calcium ions into the presynaptic neuron causes synaptic vesicles to fuse with the presynaptic membrane, so releasing acetylcholine into synaptic cleft. Acetylcholine molecules fuse with receptor sites on the sodium ion channel in the membrane of the postsynaptic neuron. This causes the sodium ion channels to open, allowing sodium ions to diffuse rapidly along a concentration gradient. The influx of sodium ions generates a new action potential in the postsynaptic neuron. Acetylcholine esterase hydrolyzes acetylcholine into choline and ethanoic acid, which diffuse back across the synaptic cleft into presynaptic neuron. In addition to recycling choline and ethanoic acid, the breakdown of acetylcholine also prevents it from continuously generating a new action potential in the postsynaptic neuron. ATP released by mitochondria is used to recombine choline and ethanoic acid into acetylcholine. This is stored in synaptic vesicles for future use. Sodium ion channels close in the absence of acetylcholine in the receptor sites.